All right, so last metabolism video before we have a very short one at the very end, and that's protein metabolism. So we've already talked about how we metabolize carbs, we've talked about how we metabolize lipids, so of course we got to go into amino acids. So we're going to talk about how we absorb them, and then the process of transamination. Why do we do that? Oxidative deamination. Why do we do that, and how do we stimulate that? So again, anabolic pathways and catabolic pathways. Anabolic, we're building new proteins, we're sticking amino acids together. This is going to be done by ribosomes, who are going to translate, right, that RNA message into an amino acid chain. And this is what we're going to do when we have high energy storages. So we have our ability to make proteins through translation. Basically, ribosomes take an mRNA molecule and use it as a template to translate from nucleic acids to amino acids, building an amino acid chain. And we can do this when we can afford it, right? When energy stores are high, so we can put our efforts into making proteins. We can make 11 of the 20 different amino acids, but the other 9 have to come from your diet. We call those essential amino acids because it's essential that you get those 9 from the food you eat. So for example, methionine and histidine, those are some essential amino acids. You have to get them from the food you eat. If you eat meat, this is not too difficult, as most meat sources have all 9 of them. But if you're a vegetarian or vegan, you may have to pay a little bit more attention to your protein sources, as it's a little harder to come by protein sources that have all 9. And you might have to eat a combination of proteins to make sure you're getting them, right? So it's not impossible, right? I'm no way saying that you cannot be vegetarian or vegan. You definitely can do that. That's entirely possible. You just have to be a little bit more mindful compared to someone who eats meat to make sure you're getting all 9 of those. Protein synthesis is stimulated by a few things like human growth hormone, thyroxine, and insulin. And if we have more amino acids than we actually need, those amino acids are used for making glycogen or fat. This is why, too, over-consuming protein, not always a good thing, right? Because it's not going to just become more protein. It can be broken down in a way that creates glycogen or creates fat, right, for fat stores. So that's our anabolic pathway, building proteins. Breaking proteins down, like when we eat the food, right, we're going to break them down into their individual amino acids, some of which can be converted into other amino acids, others not so much, right? We can also convert them to fatty acids, ketone bodies, or glucose, depending on where we are conditions-wise. So when we digest, we break proteins down into their individual amino acids. When we are fasting or starving, proteins are broken down, amino acids are used to produce ATP. And then we can also use them for gluconeogenesis to convert amino acids to glucose. So let's talk about how we get amino acids from the food we ate, right? The proteins are broken down into their individual amino acids, right? We have a bunch of enzymes that can help with that. And then we can absorb those amino acids into the small intestine cells, right? I'm not so worried about you understanding the specific transporters, but knowing that we are doing so through active transport. So active transport is being used to bring amino acids in from the food that we've eaten into the cells in our small intestine. And then facilitated transport is used to take them from inside the cells into the bloodstream. So it's really similar to carbs, right? In terms of, at least on the basic level, that active transport brings the material in to the small intestine cell, and then facilitated transport moves us from the cell into the bloodstream. Now, once we have these free amino acids, right, and we have a ton of them, right, we can use them to create something else. This is one pathway, oxidative deamination. Now, deamination, right, deaminate, we think we're removing the amino group, the nitrogen-containing group. This is what's going to really happen in the liver. And so we basically take that amino group, we strip it, right? This is going to form ammonia. Ammonia gets converted to urea, which is a main waste product we find in urine. Then we have what we call alpha ketoglutarate that can be used to make ATP. So oxidative deamination, the one thing I want you to take from that is this is our process of stripping the amino acid of its amine group, of its nitrogen containing group. We're going to form nitrogenous waste that ends up in the urine, and then we can use what remains to produce ATP. So do not worry about memorizing this. This is like, whoa. But we can see where different amino acids enter in at different points, right? Because they have a slightly different structure from each other. But the big thing that you notice about all of them is you are taking away the nitrogen containing group, the amine group, and you're producing urea, right? Urea is going to be a nitrogenous waste product that ends up in urine, right? And that all of these are going to be used to produce ATP in this case. We can also use proteins to make new glucose molecules, just like we said. This is the same slide that we saw earlier, just, in this case, we're talking amino acids. So the concept for this has not changed. You do not need to worry about the details. For the most part, it's all the same. We're just entering as pyruvate. We still go up through glycolysis to create glucose right in reverse. Now there's also transamination. Transamination is the transfer of an amino group, right, under fasting conditions. So this is basically what I really want you to get out of this is that we are going to be taking amino acids and converting them from one type to another. So here, right, we're going from glutamic acid to aspartic acid. Or here we're going from glutamic acid to alanine. So I'm not worried about you memorizing, again, any of the specifics about this. But just understanding what is transamination, we are becoming a different amino acid, right? This needs a little bit of help from vitamin B6, right? And transaminase is going to be our enzyme to help, hence the name. But so again, don't worry about all the fine detail. I just want you to remember we are taking an amino acid, we are converting it to a different amino acid.